Hi, I'm Joyce Kang, a graduate student in the Ray Chowdhury Lab at Harvard Medical School. Today, I'm excited to tell you about Symphony, an efficient and precise algorithm for single-cell reference mapping. Around the world, investigators aim to construct these large-scale comprehensive reference atlases of various tissues, with million-cell atlases emerging. So say you've invested a lot of time and effort into carefully assembling a reference atlas and annotating the cell types present. Now, you can imagine that a new investigator comes along with a smaller data set, perhaps a single 10x run, and wants to know what their cells are. The idea behind reference mapping is to be able to efficiently map this previously unseen query dataset onto the stable existing reference atlas. By mapping to the reference, we can automatically annotate the query cell types by transferring annotations from the closest reference cells. How might this be useful? Well, as an example, our lab is very interested in studying rheumatoid arthritis. By constructing a comprehensive atlas of all the cell types we would expect to find in the synovial tissue, we can then use the reference to rapidly annotate new queries. These could be synovial biopsies from new patients, single-cell data from a mouse model of RA, where we're interested in determining the most transcriptionally similar human cell states, or maybe data from an in vitro experiment, where we're interested in how cell states are transitioning. In all of these examples, we could try to redefine query cell types from scratch, but ideally, we would want to leverage the detailed and carefully constructed reference to rapidly and reproducibly annotate the query cells. Symphony offers a framework to do this. To build a reference, we start with many reference datasets, for example, multiple studies, donors, or collection sites. Here, shapes represent cell types, with colors being more fine-grained cell states. By projecting the cells into a low-dimensional embedding, like PCA, and performing de novo integration using the previously published Harmony algorithm, we can then get a harmonized reference embedding, where the cells group by cell types with unwanted technical and biological effects removed. And now this is where Symphony comes in. Symphony compression takes in the harmonized reference cells and computes the minimum information needed to map new query cells. Harmony uses an underlying linear mixture model when representing biological cell states as soft clusters. At a high level, Symphony is capturing summary statistics for the final models, representing large reference atlases in a way that is much more compressed and easier to share. The next part of the figure shows how Symphony mapping works. Starting with a new query single-cell dataset, where the cell types are unknown, we can then map the query onto the reference starting from expression by projecting the query cells into the embedding, assigning them across the soft clusters, and assigning the appropriate mixture model components to each query cell. By estimating and removing query batch effects, we can place the query cells into the same harmonized low-dimensional reference space. And then we can look at each query cell's nearby reference neighbors to automatically transfer annotations, like cell types, to the query cells. Symphony has several key properties that we hope will make it useful. First, Symphony is very fast. Even for references of 500,000 cells, mapping queries takes just seconds. This is much faster than attempting to integrate the reference and query cells de novo. Second, Symphony can handle complex study designs, like integrating references and queries across multiple technologies and species. Here, we build a reference of human pancreatic islet cells from 4 datasets, each using different plate-based technologies. And we can use it to map human and mouse cells from a new study with different donors assayed with droplet-based technology, simultaneously integrating the query cells over species, donor, and technology. These plots show the same example comparing Symphony mapping to Seurat and SC arches. Reference cells are shown as a density in gray, with individual query cells plotted on top, colored by cell type. Compared to other methods, Symphony is much faster and better able to integrate the query cells among their corresponding reference cells, both across species and donors. Finally, Symphony is very versatile. In addition to mapping to discrete cell types, it can also place query cells along continuous developmental trajectories, such as a previously published atlas of fetal liver hematopoiesis. If you're interested in learning more, check out the preprint and try out our code on GitHub. I'd like to thank my amazing teammates on this project, in particular, Ilya Korsunsky, Aparna Nathan, and of course, Shomo Ray Chowdhury. Thanks so much for listening, and I'm happy to take any questions either here or by email.